In today's lecture, we are going to discuss with you about HB electrophoresis, or in full we call it hemoglobin electrophoresis. And this hemoglobin electrophoresis is the confirmatory. This is the confirmatory test, which is done to confirm any hemoglobinopathy present. So this is a confirmatory test for hemoglobinopathies. In case you're suspecting any hemoglobinopathy involving different types of hemoglobins, this is the test of choice, and is being the test of choice for confirming sickle cell disease. So if you want to confirm sickle cell disease, or you want to confirm sickle cell trait, you can do HB electrophoresis. So in this video, we are going to see how can we confirm someone with sickle cell disease or sickle cell anemia by doing lab tests known as HB electrophoresis. So we're going to see the principle on which this HB electrophoresis works on, and the sample which we are going to use. We're going to use EDTA and coagulated whole blood. This whole blood is collected from vein, so it is venous. And when we get this EDTA blood, we are going to see that because of sickle cell, we know that sickle cell is formed due to whereby glutamate, amino acid, is replaced by valine at position 6 of beta globin chain. Beta globin. And when this one occurs, this replacement leads to, this one is the one that leads to the formation of HBS, and this HBS become less negative. It becomes less negative. So when we are measuring its electrophoretic motility, we are going to see compared to HBA, it is less negative compared to HB. So during electrophoresis, we are going to see adult hemoglobin migrates faster in electric field compared to HBS because of replacement of glutamate, which has 2 negative charge, by valine, which has a 1 negative charge. And as we are talking about HB electrophoresis, we want to look at the principle. What is the principle behind this test? As we know that this electrophoresis comes from the word movement or migration, and this one is called electrophoretic motility. Migration of charged substances according to the number of charge and mass is what we know as electrophoretic motility within an electric field. So we are going to see under the principle that if we have well mixed EDTA and coagulated venous blood, whole blood, and this blood we mix it in a cellulose. We put it in a cellulose acetate gel, maintained at a pH between 8.4 to 8.6, meaning we are using alkaline pH. And I want to tell you that hemoglobin being a protein at alkaline pH, it is positively charged. So the HB is negatively charged, at alkaline pH. So in alkaline pH, proteins including hemoglobin, they are negatively charged. So when you apply an electric field they are going to migrate from cathode toward this anode. Why? Because they are negatively charged. So when we have a sample containing HBS, which we are suspecting, and at the same time, of course, to be containing HbA, we are going to see the hemoglobin in this whole blood and coagulated venous blood is going to migrate when applied in electric field. So on application of the electric field, when electric field, if an electric field is applied, when we apply electric field at a buffer at this pH will be buffered between 8.4 to 8.6 on a cellulose acetate gel, and we apply electricity, we are going to see migration of hemoglobin, charged hemoglobin, and this migration depending on the mass of the hemoglobin and the charge. So here the factors that are playing is pH, then the mass and the charge number, whereby those which are highly charged move faster compared to those which are less charged. And we have seen that HBS is less charged compared to HbA, which is highly charged. So when we apply, so we are assuming this is an electrophoretic tank, and this electrophoretic tank is containing cellulose acetate gel. So here there is cellulose, molten cellulose acetate gel, maintained at a pH between 8.4 to 8.6. So this is cellulose acetate gel at a pH between 8.4 to 8.6, which is alkaline pH. So at this pH, if we come and apply our blood sample, this is the point of application. If this is the point of application of this sample in this cellulose acetate gel, we are going to see migration of hemoglobin depending on the mass and the charge. And we are saying HBS has less charge, it is less negatively charged, whereas HB is highly negatively charged, meaning HbA moves faster. So depending on these factors, HbA migrates faster compared to HbS. Because this is less negatively charged, this is highly negatively charged. So we are saying we have applied our sample at this point of application on the cathode. So if the sample is containing HbA, we are going to see one with the highest electrophoretic motility among the hemoglobins is HbH. HbH has the highest electrophoretic motility, followed by HbF, then followed by HbA, then the one with, we are saying HbA moves faster compared to HbS. So we are seeing HbS moves like at this point during migration in electric field, meaning we can easily separate HbA from HbS because HbS moves slowly. This one moves slowly, this one moves faster in an electric field towards anode. So we are saying HbA migrates faster compared to HbS towards anode. And remember, anode is the one which is positively charged. And remember, hemoglobins are negatively charged. Even this is highly negatively charged. So they migrate towards the positive pole, which is anode. And the migration depends on the pH, mass, and charge. So this is what we can discuss. And I want you to note, we have seen that HbF have highest electrophoretic motility. Electrophoretic motility, whereas then it is followed by HbF and Hba. Then we are going to tell you that HbS and Hbd have the same electrophoretic motility. They have the same. So even if I have Hbd in the sample, it is also going to migrate at the same level. So this one is Hbd. So Hbd and Hbs have the same. They move at the same point in alkaline media. So how can we separate Hbs from Hbd? In case a sample contains both, we can separate this by lowering the pH at 6. So when we run this test at pH of 6, so these ones are separated at pH of 6. Separated by lowering the pH of 6, slightly acidic media. We are able to separate Hbd from Hbs. That is very important to note. And lastly, the ones with the lowest electrophoretic motility is, we are having two of them. They also have the same, whereby we have Hbe and Hbc. So Hbe and Hbc, they also have the same electrophoretic motility, and the last one is also Hba2, has the lowest electrophoretic motility. So all these are multiple choice questions. They can easily ask you which of the following hemoglobins have the highest, that is HbH. HbH has the highest electrophoretic motility. It is HbH, not HbF. Then followed by F and A, as we are seeing on this electrophoretic tank. Then followed by D and S, which have the same electrophoretic motility, and we have seen can be separated by doing the pH, the test at a pH of 6.0. Then the last ones with lowest, we have also HbC and Hbe, which also have the same electrophoretic motility, and lastly Hba2. So this is what we can see under electrophoretic Hb electrophoresis, whereby the principle remains the same. When you get your well-mixed EDT and coagulated blood, you mix it well in a cellulose acetate gel and you brought it at a pH maintained 8.4 to 8.6, that is alkaline pH. Proteins exist as negatively charged. So when we apply the electric field, we are going to see these negatively charged molecules of protein called hemoglobin migrating from cathode towards anode where it is positively charged, depending on the mass and charge. So the one which is highly charged migrates faster towards anode compared to the one with less negatively charged. And that's why we say Hba, which is, we are just comparing that Hba, which is highly negatively charged, migrates faster than Hbs, which is less negatively charged, towards the anode. And this is what we can discuss under the principle of Hb electrophoresis. So hope you have gained a lot from this, whereby we have looked at that this is the confirmatory, and we have seen why HbA moves faster or migrates faster than HbS. Thank you so much for learning with us. May God bless you.